Welcome back to this last video on the endocrine system. And for this one, we're going to return back to where we started from. And that is looking at more hormones that are unrelated to the pituitary gland. So we're going to move to the pancreas and then also talk a little bit more about the adrenal gland. So your pancreas is in your upper abdomen. It actually sits posterior to your stomach. It's sort of in the midline with the head of the pancreas emptying into the first part of your small intestine and the tail of the pancreas going to your left upper quadrant and ending at the spleen. If you were to take your pancreas and in the midline, essentially do a sagittal section, this is what it would look like. You would notice that you had a big duct in the middle, and there were glands, in this case, these blue raspberries, which are opening and dumping their secretions in the glands. So this is the exocrine portion of the pancreas, and that's the part we'll learn about when we learn about the digestive system. The part that we're going to talk about now are these red raspberries, which are dispersed throughout the pancreas. And these are known as islets of Langerhans. The little islands of different types of cells, they're these pale areas interspersed among the normal exocrine tissue. And there are at least 4 different cell types, each of which produces a different substance in the islets of Langerhans. But we're going to talk about 2 of them. We're going to talk about the 2 pancreatic hormones that are most important for glucose homeostasis. And those 2 hormones are going to be insulin and glucagon. So most folks know a little bit about insulin and really have probably never heard of glucagon. So before you continue, I want you to stop and think what you know about insulin and diabetes and kind of write that down as we go along this part to see how much of what we're learning is actually different than what a lot of the lay people think is true. So go ahead and write down your thoughts on diabetes and insulin, anything you might know, like what are the problems with blood sugars, with diabetics, what other problems might they have that you know about? Okay, so let's get started and let's start with glucose storage and understand what happens with glucose so that when I use certain terms, you will know what they mean, or at least you can come back and review. So this is your liver in your right upper quadrant, and in the cells of the liver, inside the hepatocytes, a couple of different things can happen. If we have glucose in our blood, we can take that glucose and make a long chain of that glucose, a molecule with a long chain called glycogen. That process is called glyconeogenesis. Okay, neo means new, genesis is to create, and we're creating new glycogen. And our livers normally store about 48 hours worth of glycogen. So if we're not eating, this can be broken down back into glucose. And that process is called glycogenolysis, lysis, where we're taking the glycogen and cutting off the glucose molecules. Now, in addition to this, say for instance, all of our glucose stores were already full, then the glucose, if we needed it, immediately could be broken down into acetyl-CoA through the process of glycolysis, and the acetyl-CoA could then enter the Krebs cycle, and we can make a lot of ATP from the Krebs cycle and electron transfer chain. And similarly, we could take those products and make glucose out of it through the process of gluconeogenesis. So that's what happens in the liver. But another tissue that's super important for storing glucose when we have too much of it is our adipose tissue. So we've already made all the glycogen we need, that any other extra glucose that enters the liver can be sent to an adipose site. And in the adipose site, it's going to go through a series of chemical reactions and eventually form a triglyceride, which is three fatty acid chains attached to a glycerol molecule. And then if we need energies, we can take that triglyceride, cut the free fatty acids off of it, which actually can give us some ketone bodies, and then we will go back and we will, through more chemistry, end up with a glucose molecule, because we're going to take that we're going to take the glycogen and break it down back into glucose coming out of the adipocyte, just like I showed in the hepatocyte, that glycogenolysis. All right, now that you understand what we can do with glucose in our body, let's talk about glucagon and go back to those islet cells. So here is the cartoon drawing of an islet cells, and the alpha cells are these pink cells, which are mostly among the periphery. And this is a photomicrograph done with special histological stains, which are staining for the presence of glucagon. So you can see that they're mostly around the periphery of the islets. So yes, you do need to know pancreas. Yes, you do need to know islets of Langerhans. Yes, you do need to know alpha cells is where glucagon comes from, and that insulin's going to come from a different cell type. So here's what happens. When you have a low blood glucose, your pancreas gets the signal, and the alpha cells, which actually already have some pre-formed glucagon in them, will then release the glucagon into the blood, and you will achieve normal blood glucose levels later on. If you continue to have low blood glucose afterwards, then the alpha cells will continue to make some more glucagon. So what are the effects of glucagon? Well, going back to the glucose metabolism slide, it stimulates gluconeogenesis. In other words, if we got some acetyl-CoA floating around, we're going to make glucose from it. In addition, it stimulates liver glycogenolysis. In other words, the glycogen in the liver is going to be chopped up to release its glucose molecules. So it can use amino acids if needed. So in other words, if you don't have glucose floating around and you don't have it in your adipose tissue and you don't have glycogen in your liver, then it can use amino acids and break them down, get your acetyl-CoA, and then get glucose from that. And we really don't want to be breaking down muscle if we don't have to. Okay, this picture right here is kind of the sparks note of the two big pictures that I showed on a couple of slides ago. All right, so glucagon stimulates fat breakdown as well, okay, because in order we can, when we break down triglycerides, we can then have the glycerol, which can be used to make glucose from it. But a side effect of when we break that down is the production of something called ketones, which actually will change your pH in your blood. So to have massive ketone production is not a good thing. All right, so in contrast to glucagon, let's talk about insulin. Insulin comes from the beta cells in the islets of Langerhans in the pancreas, and the beta cells are these golden cells. So as you can see, they are pretty dispersed throughout the entire islet, except that they tend not to be in the periphery. I want to discuss this picture just for a moment because this shows what happens to blood glucose levels if, for instance, you were to drink, say, the syrup that a soda is made with. You're drinking a glucose load, which essentially is like drinking Fanta syrup without the carbonation in it. And what happens within about 20 minutes, your blood glucose level, represented here by blue, is going to start increasing. And within 20 minutes of that happening, your beta cells are going to start kicking out the preformed insulin so that you can see the blood concentrations of insulin are going to be mirroring the blood concentrations of glucose. And then once the glucose levels start falling down because the insulin has dropped the glucose levels, then the insulin release starts plummeting as well. And within 2 hours after a glucose load, your blood glucose level should be back at baseline. You should have normal blood sugar levels at this, equivalent to your normal fasting blood sugar levels. So the threshold for releasing insulin actually is in the upper normal range. So you don't have to be super elevated. The fact that blood glucose levels are going up within the normal range is enough for these beta cells to start kicking out their insulin. And the half-life for insulin is only about 6 minutes, which means you're not going to be getting hypoglycemic as a result of kicking out insulin. So when we have high blood glucose levels, it's the beta cells of the pancreas that kick out insulin. And then we're going to learn on the next slide what actually happens to the blood glucose to make it return to normal. A lot of people think it's the insulin that attaches to the glucose and breaks it down in the blood, and that's how blood glucose gets normal, and that is nothing related to the truth. But before we get there, I want to talk about how insulin is formed in the beta cells. Okay. So when we have elevated or rising levels of glucose, this rapidly increases translation of insulin messenger RNA. So remember that transcription translation part. So this rapidly increases the production of the amino acid chains for insulin at the mRNA and increases the transcription of the gene. And what gets formed is something called pre-proinsulin, which has 3 amino acid chains attached to a signal peptide. And this is an inactive precursor that's formed and is found in the rough endoplasmic reticulum inside the beta cells. And then if you remember, the rough endoplasmic reticulum is going to do some packaging and it's going to send stuff to the Golgi apparatus for further modification. And what happens in the Golgi apparatus is the pre-proinsulin loses its signal peptide and becomes a compound known as proinsulin. This is still an inactive precursor. Now, what happens to the proinsulin is that we form bonds between the alpha and the beta chains on the 2 ends, and that turns it into this loopy kind of structure where the blue parts actually are what's considered to be insulin, and those 2 parts are attached by this C-shaped golden part, which is called the C-peptide. And this gets stored, if it's not immediate needed, it'll get stored in granules. And then once it gets its metabolic signals, including some vagal stimulation, that C-peptide is cut off of it, and you are left with just the insulin fragment itself, which is released through exocytosis into the blood. So what is very interesting is I told you the half-life of the insulin molecule is only about 6 minutes, which means however much is released, half of that is gone in 6 minutes. And another half would be gone by the 12-minute mark. So 75% would be gone by 12 minutes. And then 87.5% would be gone by 18 minutes. And so it makes it kind of difficult to measure insulin levels, and are people kicking out normal amounts of insulin when they're given this glucose load. But what we have learned is this C-peptide has a much longer half-life, and it's a good indicator of insulin production, especially in early cases of diabetes. You can realize that the person is kicking out no insulin or the person is kicking out lots of insulin. So what does insulin do? Well, it's kind of like a key that when it goes in its slot, opens the door, next door. So insulin has its receptor on its certain target tissues, and we'll talk about what those are in less than a minute. And when insulin binds to its receptor, those cells have a transmembrane protein that's called a glucose transporter. In other words, this channel will open up when insulin is attached to that cell. So insulin attaches to the cell, the glucose transporter channel opens up, and glucose that is in the blood can then go into that cell. So what are the target tissues? Well, there are 3 of them. First one is the liver, which is what's being shown in the lower left quadrant, where when glucose goes in, it can be made into glycogen and stored. Second one is adipose tissue, where we showed it can be stored as a triglyceride. And the third one is muscle. Your myocytes will take the glucose and use them to create ATP. In addition, when glucose is sent to the adipose tissue in somebody who has normal blood sugar levels, this can stimulate the hypothalamic satiety center, so hopefully you will quit eating. However, we know that there are lots of hormones that will work on your satiety center, and so this is not absolute. So at this point, I would like you to pause and think about the scales between insulin and glucagon and glucose, and talk through the process of what is happening. And don't forget to use those words: glycogenolysis, gluconeogenesis, glycolysis, and make sure you understand those terms before you continue on. All right, thank you for doing that exercise. Now I want to diverge a little bit before I get into diabetes and talk about something called the metabolic syndrome. And basically the metabolic syndrome is when we look at 5 risk factors. If you have 3 of the 5, then you are diagnosed with metabolic syndrome. And the 5 risk factors are high levels of triglycerides, low HDL cholesterol. HDL cholesterol is the good kind of cholesterol, and if it's low, that means it's not there to protect you. Visceral obesity, which means you are shaped like an apple, and you carry your adipose tissue in your trunk. Insulin resistance, which I will talk about, and high blood pressure. Now, if you have any 3 of these, then you are diagnosed with metabolic syndrome. And metabolic syndrome means you are at much higher risk for diseases such as heart disease or diabetes and stroke and dementia, as well as things such as polycystic ovary disease, which makes it impossible, nearly impossible, to be getting pregnant. And non-alcoholic fatty liver disease, which we now know over years can turn into cirrhosis. So, and what else is interesting, folks with metabolic syndrome, because they're at risk for all these bad diseases, a mere lack of sleep really upsets their physiology and increases the risk of sudden death. So, who has metabolic syndrome? Well, it's all very easy for all of us to point the finger, and y'all can point the finger at me and go, oh, your bad habits, you're eating sweets all the time, or junk food, or having sodas, and you're not exercising. But I want you to realize not everybody who looks like that has bad habits, and not everybody who doesn't look like that doesn't have bad habits. Think of the college folks that you know who sit there eating their junk food every weekend and their sweets when they are stressed and having sodas. And oh, yes, I'm too tired to walk across campuses. College campuses are so big. And like, you'll circle the parking lot 3 times rather than park a 3-minute walk away from the door. Okay. So even at a healthy weight, even if you are skinny, you can have metabolic syndrome. Okay. So you cannot prejudge people. So that brings us to insulin resistance, because this is definitely something that people with metabolic syndrome, as well as other people, have. What happens is When you have this lack of activity, especially, that's the biggie, your body, the cells in your muscles, your adipose tissue, your liver, those places where the insulin is the key for uptaking the glucose, they start ignoring the signal. And they go, dude, you're never exercising. I don't need this. No, we're not doing this. Those days are done and over. Haven't you heard? COVID, nobody's exercising anymore. Okay. And so then what happens is your body's going, oh my God, blood sugar's still high. And it starts kicking out more insulin. And what happens is because of the insensitivity, the resistance to insulin, your insulin levels go higher and higher and higher. Okay. And then because you have high glucose levels, because the insulin's not working, then you feel crappy. And so then you eat some more, you eat bad carbs, and you make more insulin. Your cells are resisting that insulin. You're going to store more of that as fat. You're going to feel crappy. You're going to eat some more. And you see you're in this little vicious circle of promoting your insulin resistance. When what you need to do is high intensity exercise, because this is the magic key that opens the plasma membrane so the blood glucose can enter the cell, the muscle cell. Okay. And then you will start losing your insulin resistance. Your body will quit making as much insulin, and you'll start kumbayaing in homeostasis all over again. All right. So what happens with high blood sugar, hyperglycemia? Well, you have beta cell dysfunction in that either you have no insulin being produced, or you have what we call impaired insulin secretion, because even though you may be kicking out insulin, even though you may be kicking out massive amounts of insulin, your insulin resistance. So for whatever reason, the insulin is not doing its job. You also can have hyperglycemia from increased glucose being produced out of the liver, and also from adipose tissue with its breakdown of triglycerides. So there's a lot of reasons why you can have a hyperglycemia or increase in blood glucose. So this brings us to the diagnosis of diabetes mellitus. So there's a couple of different types of diabetes, and the type that most people are talking about when they just, well, that everybody's talking about when they just use the word diabetes is diabetes mellitus. And here there are at least two different types. And when I say different types, we are taking out the diabetes of pregnancy, although it is associated with a much higher incidence of diabetes mellitus. So type 1 diabetes is when the pancreas fails to produce insulin in response to elevated or rising blood sugar levels. So there is no magic key to put in the insulin receptor to let the glucose enter the cell. And this most often now we know is an autoimmune result, and very often follows some type of viral infection that affects your pancreas. And it was previously known as juvenile diabetes because it was more common in kids than adults. Type 2 diabetes, which used to be called adult onset, we no longer use that term because we start diagnosing this in kids in elementary and middle school, which just boggles the brain. And type 2 diabetes is where the beta cells fail to respond to and release the appropriate amount of insulin for what the blood sugar is. In other words, you're going to kick out some insulin, and a lot of these people have elevated levels of insulin because they have the insulin resistance from lack of activity. So a lot of people can work on type 2 diabetes with dietary changes and exercise changes, especially if they're in that pre-diabetic phase. So when the cells can't absorb the glucose, whether you have type 1 diabetes or type 2 diabetes, then the body is going to shift to fat or protein for its energy usages. So here's just a little graphic showing you that the vast majority of people, more than 90%, are type 2 diabetics. What risk factors are, it's in about 9% of the population, and 25% of the folks with diabetes are unaware that they have diabetes because they don't go for their routine screenings. And there are lots of different parts of diabetes management. Number one is going to be, for type 2 diabetic, is going to be to increase physical activity, and for both of them is proper diet. Type 2 diabetics, some can use medication, but all type 1 diabetics are going to need insulin injections or the insertion of insulin pumps. When it comes to signs and symptoms of diabetes, I've given you this slide with only 2 pictures of it because I want to explain how those 2 things happen. A lot of people know that the diabetics, they know they're drinking a bunch of extra fluid, and if they think that's why you're peeing. Well, it would make sense. If you drink a lot, you're going to pee a lot. But let me show you how this really happens. Diabetics have elevated blood sugar levels, and what happens is that elevated blood sugar goes into the kidney where it gets dumped out in the tubules, which is where the urine comes from. And in order to go back into the blood, it needs a protein carrier. And your body only makes enough protein carrier for normal levels of blood sugar, which means only the normal amount of blood sugar gets sent back into the blood and the rest stays in your kidney tubules. And that glucose in your kidney tubules has a huge osmotic pull. It sucks in the water, so the volume of urine increases. And that's what causes the frequent urination. And so because you are urinating, you're losing a lot of fluid, which then makes you thirsty and makes you drink more fluid. So oftentimes the only symptom is people drinking extra fluid. And so all you people think, oh my gosh, I need to drink a gallon or 2 of water a day. Okay, you don't, okay? When that amount of fluid you need in your everyday, you get to include the fluid that's in all your foods, unless you're just eating beef jerky all day. There's lots of water in your food stuff. So you kind of should be honest with yourself. Are you drinking more water than what you need? And are you urinating more often than your peers are? Other signs and symptoms of diabetes is they're often very fatigued. Because diabetes is associated with certain processes that affect blood vessels and affect nerves, you can get numbness and tingling in your feet. The blood vessels can change your vision. Oftentimes your eye professionals will be the ones to diagnose diabetes in people who don't go to other healthcare professionals. Sexual dysfunction. This is a major reason why you want your diabetes under control, gentlemen, or you won't be able to get that erection. So those are the 2 I want you to focus on. I want to discuss something called hemoglobin A1C, or glycosylated hemoglobin. And this is now how we kind of monitor people's blood sugar levels, especially with diabetes. It used to be that we would have people, you know, and they're checking their finger sticks for their blood sugar levels and writing down whether or not how much insulin they need to take. And then they take all their blood sugar levels and they go to the doctor and show it to the doctor. Okay, but people lie. It's that simple. People with diabetes, they want to go a party, they're going to eat the birthday cake, they're going to eat the wedding cake, they're going to do whatever they want to do. They know they're not exercising, the blood sugars are high, maybe they're at 200, they're going to write down that they were 130, right? Because they don't want to feel bad when the doctor can notice that they're not doing what they're supposed to be doing. So this was a great discovery. What we discovered is glucose actually attaches to hemoglobin, and that's what's called glycosylated. And the rate of this depends on the concentration of glucose in the blood. So the lower, more normal the glucose levels, there's not very much glycosylated hemoglobin. Now, if you have this feast, or if you're feasting all day and your glucose levels are high for prolonged periods of time, then you're going to have a higher percentage of glycosylated hemoglobin. So by glycosylated hemoglobin, what I mean on the hemoglobin molecule, there's this amino group to which the glucose can attach, okay? And this is an irreversible bond. So even if you starve yourself and are hypoglycemic, this bond will not break. It is always there. Now we know that this hemoglobin will circulate in the blood for about 120 days. So we know what percent, you know, like so 1/120th, less than 1% of the blood is new blood from that day, and less than 1%'s been around for 120 days. And so these serum hemoglobin A1C levels indicate what has been happening to the glucose values in your blood for the previous 120 days or 3 months. So you don't have to bring in 3 months worth of data for the doctor. They just do a hemoglobin A1C and they know what's been happening for the previous 3 months. And so normally it's less than 6% that is glycosylated, and 7% is the cutoff for somebody that's diabetic. Okay? So there's very little wiggle room in there. And that brings me to the greatest Canadian invention of them all, insulin, which happened in 1922 when doctors were, well, actually it wasn't doctors that discovered it, but it was a great thing that happened in Toronto, at the University of Toronto, a lab associated with that. And this young 14-year-old named Leonard Thompson, I know he doesn't look 14. Back in those days, people with diabetes would be put on 500-calorie diets, and they were lucky if they lived for 4 to 6 months. This was such a great invention. The entire medical report in the journal Science took maybe 1.5 pages, which is I use as proof. You don't need a 10-page essay to do good work. And this was given, the Nobel Prize was awarded, as you might expect. But the government of Canada did something in that they recognized this could save lives worldwide, and they worked with the drug companies and made the drug available within a year of its discovery. And they made it available worldwide, royalty free. And since then, it has saved millions of lives. We no longer use insulin from cows and pigs like we used to, and now most insulin, well, we do for some people, but most insulin is recombinant DNA based. All right, enough for diabetes at this point. Anybody stressed? Okay. I put this picture here because this picture I found on the front page for Paleo Diet Lifestyle, and I just thought, isn't the whole point of Paleo Diet not to be stressed? But yeah, when you're trying to follow too rigid a diet, you're going to be stressed. So when you are stressed, here's what happens in your body, putting lots of things together. The adrenal medulla, as you know, is going to release catecholamine. So you're going to have elevated epinephrine and norepinephrine levels. In addition, the adrenal cortex is going to release glucocorticoids. So your cortisol level is up, thereby throwing your body out of whack. You're also going to have neural input effect on your hypothalamus. So blood glucose levels are going to go sky high. Stress, diabetic, bad combination. In addition, remember that cortisol suppresses your immune response and affects many organ systems. So your immune response, so think about it, if you're diabetic and you're under stress, severe stress, say it's finals week, or say you got a diagnosis of cancer, your immune response is being suppressed. So you have bacteria that your macrophages aren't looking to get rid of because they're being suppressed. They're being told, go on siesta. And your blood glucose levels are high, which is kind of like those bacteria are now at a freaking Vegas buffet eating 24/7. There is no lack of understanding why diabetics have so many infections at times of stress. This is just a cute cartoon. I suggest you stop the page and look at it because it's pretty simplistic. And now let me show you what happens to all these hormones in your body that we have learned about at times of stress. Aldosterone levels are going to go up. So as you know, that'll decrease your urine output, which increases your blood volume, which means your heart has to work harder. Antidiuretic goes up, which means once again, blood volumes go up. So once again, your heart will have to work harder to pump all that blood volume because those blood volumes are going to increase your blood pressures. In addition to that, the increased levels of epinephrine and norepinephrine as catecholamines coming from the adrenal resistance, they cause your peripheral blood vessels to vasoconstrict, and that also increases your blood pressure, which is now sky high. The glucagon increases your blood glucose levels, increasing your risk of infection, and your increased beta endorphins increases your pain suppression. So if you do have an infection, you're not feeling it. Also affects your gonads. I just don't want to talk about that. All right, so do you have any stress? Well, let's look at the common stressors and see. Family relationships. Yeah, I'm pretty sure no teenager has ever argued with a parent. So no, I would say you probably grew up your whole life without any stress with your family. You don't have any disagreements, or not very many, and you kind of work them out because you like to talk them out, and so everything's copacetic there. Money worries, I don't know a single student with money worries. Need to succeed, well, of course you're going to succeed. That's why you're taking this class, right? Exams, well, they're a piece of cake. Y'all know how easy the exams in this course are. Friends, well, your friends are always there for you, right? Especially when you need them. They're never going out on a date with a new hottie instead of coming to you in your time of need. Yeah, I knew you had the best friends. You don't have to make any decisions, much as like, what am I going to do with the rest of my life? No. And as far as new situations, no. You understand everything that's going on. You're never going to need another new job. You are set for life. So you have no stressors. But let's assume for a moment, perhaps you did have a stress or two. And basically, that's going to put you in the fight or flight where you have to, for short-term kind of stuff. And so for these long-term stressors, basically the magic word is you've got to learn some mad coping skills. In other words, every single day, you have to proactively take 20 minutes and do personal de-stressor. And before you say, I don't have time for this, what you'll realize is the fact of de-stressing and recentering yourself makes you much more effective at, for instance, studying. Okay. Now, when I say de-stress, I don't mean take a 4-hour escape from reality. I mean literally take a power nap for 20 or 30 minutes max. Set your alarm, okay? If you take a power nap for 20 minutes, you're not in that deep REM thing, you'll be able to wake up. If you set your alarm for 60 to 90 minutes, you're right in your deepest sleep and you're going to be super groggy and not able to focus for a whole nother hour afterwards. Or watch a half an hour of whatever your guilty pleasure is on HBO or Hulu or, Amazon Prime, whatever you're doing. Read a book. Take a bubble bath. Don't drink wine if you're going to study. And alcohol, by the way, really doesn't relieve stress. It introduces a whole bunch of other stuff. A lot of fights have happened after alcohol, so don't consider it your de-stressor, nor should you consider having sex a de-stressor either, because it does not. You may think it does, but it does not. So figure out, if you need to read the Bible, if you need to go for a walk on the beach, if you need to walk your dog, if you need to go for a run. Because if you don't figure out how to de-stress your life now when you're young, it's only going to get worse. And eventually you are going to end up with dis-ease. Dis-ease meaning you are not at ease. In other words, you have not de-stressed. And yes, this will manifest in a plethora of diseases. So, chronic stress leads to significant hormonal imbalance. So that directly can lead to a lot of diseases and trigger flare-ups and things such as asthma and arthritis and other lung diseases and cause heart attacks. So don't kid yourself that I can handle this. God never gives us more than we can handle. Okay? Don't believe those platitudes. Okay. So, quick review. 3 different stimuli to manufacture and release. Humoral stimuli, which are blood concentrations, neural stimuli, which is the adrenal medulla and its catecholamines, and hormonal stimuli, which is everything else. And don't forget negative feedback loops. Make sure you can do negative feedback loops. We're pretty much almost, not the positive feedback hormones, but the negative feedback hormones that we have learned. And with that, if you're feeling stressed, just look at these pictures of wonderful macarons from Paris. Okay, I will see you soon. And thank you so much for all your hard work in the endocrine system.